Good morning. It's February 27th, and this is your daily brief in science. Here's everything you need to know. A recent study from the University of Colorado Anschutz Medical Campus has unveiled important insights into the behavior of immune cells, specifically macrophages, in response to bacteria. Published in the journal Nature on February 26, 2025, the research illustrates a crucial difference in how macrophages react based on whether they ingest live or dead bacteria. When macrophages consume live bacteria, they initiate an inflammation response. However, ingesting dead bacteria does not trigger this reaction. This distinction is significant because dead bacteria contain a molecule known as CAMP, which signals to macrophages that the bacteria are no longer a threat, helping to regulate inflammation. Additionally, the study found that macrophages convert the consumed bacteria into essential nutrients for protein synthesis and energy, rather than simply storing them in specialized compartments as was previously understood. The research identified the mTORC1 protein complex as a key regulator in how macrophages utilize these nutrients, providing new insights into the control of inflammation. This understanding is vital, particularly since chronic inflammation has been linked to various health issues such as cancer, long COVID, and chronic fatigue syndrome. Co-senior author Angelo D'Alessandro, Ph.D., highlighted the relevance of this research in the context of increasing antibiotic-resistant bacteria. He suggested that insights into natural inflammatory responses could inform future treatment strategies. The collaborative effort of this study included contributions from researchers at CU Anschutz, Charité Universitätsmedizin Berlin, and the University of Bordeaux in France. Dr. D'Alessandro cautioned that antibiotic-resistant bacteria are expected to become more prevalent in the coming decade, making the understanding of immune responses even more critical. The findings from this research could pave the way for new therapies aimed at managing inflammation, a growing concern in today's healthcare landscape. Overall, the study emphasizes how the type of bacteria consumed by immune cells influences their composition and behavior, opening up potential therapeutic avenues. A new artificial intelligence approach is making strides in identifying genes associated with neurodevelopmental disorders, including autism spectrum disorder, epilepsy, and developmental delay. Researchers have developed this innovative method to enhance predictive models by utilizing over 300 biological features, focusing on gene mutation intolerance and interactions with known disease-associated genes. The AI models aim to validate emerging genes from sequencing studies that currently lack sufficient statistical evidence of their involvement in these disorders. This research was published in the American Journal of Human Genetics on February 26, 2025. And involves a collaborative team from prestigious institutions such as Baylor College of Medicine and the University of Melbourne. Funding for the study came from organizations including the NIH and the Hevolution Foundation. The primary goal of this research is to improve molecular diagnosis and targeted therapies for neurodevelopmental disorders, addressing a significant gap in genetic understanding. Dr. Ryan S. Deinze, the first author and assistant professor at Baylor College of Medicine, noted that many patients still do not have genetic diagnoses, indicating the potential existence of undiscovered genes. The AI models revealed that the top-ranked genes identified are 45 to 500 times more likely to be supported by existing literature compared to lower-ranked genes, thus enhancing their credibility. This computational tool is crucial for advancing the genetic understanding of neurodevelopmental disorders, which is essential for accurate diagnoses and targeted therapies. Traditionally, gene discovery involved sequencing genomes of affected individuals and comparing them to those of unaffected individuals. However, this study introduced a complementary AI-based method to identify patterns among known genes. Researchers at the Massachusetts Institute of Technology have made strides in health technology by creating an elastic fiber computer that can be woven into clothing. This innovative development allows for real-time monitoring of health and activity, providing alerts for potential health risks. The construction process of these thread-based computers involves folding chips into a conducting box, connecting them with copper wire, and encasing them in a protective plastic tube. This tube can then be covered with fabric, simplifying the integration of sensors compared to existing electronic fabrics. However, further validation of their durability during everyday washing is necessary, as initial tests have only assessed water resistance. This technology offers a significant advantage over traditional wearables by enhancing the ability to track physiological data across larger areas of the body. In initial tests, the fiber computers demonstrated a 70% accuracy in recognizing different exercises, which improved to nearly 95% when multiple units were used together. Researchers successfully stitched the threads into clothing and identified movements such as lunges and squats. Currently, the technology is being tested under extreme conditions as part of Operation Nanook, led by the Canadian Armed Forces. This initiative aims to monitor body temperatures and movements in challenging environments. Additionally, the threads could assist in personal safety by detecting falls, particularly for vulnerable individuals, as noted by researcher Theo Hughes-Riley. These fiber computers operate through a textile network, relaying health data to smartphones for continuous monitoring. Each thread is equipped with 256 kilobytes of onboard memory, sensors for temperature, heart rate, and body movements, along with Bluetooth connectivity. Future developments are expected to enhance the functionality of these fiber computers, potentially revolutionizing everyday apparel with integrated health monitoring and environmental responsiveness. Lead researcher Yoel Fink suggests that this technology could pave the way for health monitoring applications that exceed the capabilities of current smartphones. A new study published in Nature Genetics has shaken the foundations of our understanding of tumor formation in neurofibromatosis type 1, or NF1. Conducted by researchers from the Wellcome Sanger Institute and UCL Great Ormond Street Institute of Child Health, this research reveals that genetic changes alone are insufficient to explain how tumors develop in this condition. The study analyzed nearly 500 tissue samples from a child with NF1 and additional samples from 9 adults. It found that the loss of NF1 gene function was not confined to tumors, indicating that mutations alone do not trigger tumor formation. NF1 affects around 25,000 individuals in the UK, leading to symptoms including brown skin patches and various tumors, some of which can become cancerous. The researchers discovered that the nervous system exhibits a higher frequency of mutations, providing insight into why tumors frequently arise in this area. Their findings suggest that genetic alterations associated with tumors are also present in normal tissues, pointing to a more complex model of tumorigenesis. This research signifies a shift in understanding tumor development in NF1, with implications for improving monitoring and early detection strategies for affected patients. Dr. Thomas Oliver, a co-author of the study, highlighted the need to investigate additional factors influencing tumor development to enhance personalized care for NF1 patients. Dr. Oliver also noted the surprising extent of genetic changes found in normal tissues, which contradicts traditional views of tumor development. The findings may also suggest that similar mechanisms could exist in related genetic conditions, potentially benefiting a broader patient population. Advanced sequencing technology played a crucial role in this study, allowing researchers to observe genetic changes at a higher resolution across the samples studied. A remarkable discovery in Siberia has revealed a tiny worm, known as Panagrolimus colimensis, that has been revived after being frozen for approximately 46,000 years in permafrost. This finding showcases the extraordinary resilience of life and raises intriguing questions about survival in extreme conditions. The specimen was found about 40 yards underground, preserved in a state called cryptobiosis, where its metabolic activity completely halted. Remarkably, after thawing, the worm was able to resume normal activity and even reproduce. This research, published in PLOS Genetics, highlights the significance of studying cryptobiotic organisms and ancient permafrost samples. Understanding the cryptobiotic process could lead to advancements in several fields, including organ transplants, space exploration, and biotechnology. The worm possesses specialized proteins that function as biological antifreeze, suggesting potential applications for human organ preservation and long-term space travel. Genetic analysis indicates that this newly identified species shares similarities with the well-studied nematode Caenorhabditis elegans, but also features distinct survival traits. while other organisms like tardigrades and brine shrimp exhibit cryptobiosis. The longevity of this nematode's frozen state is unprecedented. The Siberian permafrost acts as a natural time capsule, with radiocarbon dating confirming the worm's age, linking it to the Ice Age alongside woolly mammoths and early humans. Dr. Philippe Schiffer, the study's lead researcher from the University of Cologne, expressed amazement at the worm's ability to resume life after such an extended dormant period, challenging previous assumptions about biological survival limits. This research opens up new possibilities for understanding survival strategies in other extreme environments, such as Mars or the icy moons of Saturn. Future studies may focus on isolating the genes that allow the worm's cells to withstand freezing, which could have significant implications for food preservation and biomedicine. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.